Hmm. Well, good afternoon, everyone. My name is Sharon Rounds, and I'm the program director for Advance-CTR, which is the NIGMS-funded research infrastructure building grant for the state of Rhode Island. Advance-CTR has had a translational research seminar series now for this is the 7th year, and this year we're featuring distinguished clinical and translational research scientists who will be speaking monthly on Thursdays from noon to 1. This week, we're really pleased to have Mukesh Jain as our distinguished clinical translational research science speaker. Dr. Jain is the 8th Dean of Medicine and Biological Sciences at Brown University. And he came here on March 1st of 2022. I'm happy to say that he's had a tremendously positive impact on our community in many, many ways since his very recent arrival, and we're really thrilled to have him here with us today. Mukesh is internationally recognized for studies that established a central role for Kruppel-like factors, which is a family of DNA transcription factors in cardiovascular biology, immunity, and metabolism. Prior to coming to Brown, he was Chief Academic Officer at University Hospitals Health System in Cleveland and Vice Dean for Medical Sciences at the Case Western Reserve University, and Harrington Endowed Scientific Director of the Harrington Discovery Institute. His numerous clinical and academic contributions have been recognized through membership in the National Academy of Medicine, and he served as president of the American Society for Clinical Investigation. We're just really pleased to have you here with us today, Mukesh, and you'll be telling us about your work on new insights into vascular thromboses. So welcome.
Well, thanks very much, Sharon. And I'm just going to do a quick sound check. Can you hear me okay? Okay. All right. Well, good afternoon, everyone. Great to be with you. And Dr. Rounds, thanks for that very kind introduction and the opportunity to be here. And really to congratulate the group. You know, Sharon is the leader, but all of you are the reason why Advance-CTR is such a successful effort that really enriches our community. So I just want to give a shout out to you and the many other members of Advance-CTR that make this possible. So that's one. Now, the other sort of short preamble is Dr. Rounds asked me to say a few words about how I became engaged in translational research. And so I'll be very brief about that, and then I'll go into the seminar. But I, truth be told, did not have any aspirations to do research and came into research quite late. In fact, it was, you know, I'd finished medical school, had completed my clinical training in internal medicine at the Beth Israel Hospital in Boston. And in the second half of our third year, we were allowed to have 6 months to engage in research. And I had some wonderful mentors with whom I've worked, enjoyed some success in a relatively short period of time. And it led to a first author paper in a journal in the cardiovascular world that is thought to be quite good, called Circulation. And that gave me some confidence. And then I spent a year a year became 3 years, working with really an amazing mentor, the late Edgar Haber, who was head of cardiology at the Mass General and then a professor of biological sciences at Harvard. And Dr. Haber and that experience, again, 1 year that became 3, really instilled in me, frankly, the courage and the confidence and the know-how how to be an investigator. And the passion that he exhibited and the success that his mentees enjoyed really made it a fun and viable, a fun environment and a viable career path. And so that really changed the trajectory. I mean, I really went to do internal medicine cardiology with the plans to go into practice with my brother down in Virginia. And it completely changed my career trajectory. And I haven't looked back and stayed in academia. And as I always say to young folks, it is just a great career path to have the opportunity to integrate science and clinical medicine and clinical care. The diversity of experience is fabulous, and the opportunity to work with like-minded colleagues across the community, across the globe, is a real pleasure and privilege. So that's my short preamble, Sharon, on how I got into translation research and life as a physician scientist. I hope that works. Okay. I got a thumbs up from Dr. Rounds, which is pretty good because she's a tough critic. So with that, I'll get started. And here's the overview. I'll start with a couple of slides that are not related to Kruppel-like factors, but I wanted to share with you the thought process and what we were looking for and how that led to the identification of Kruppel-like factors as essential regulators of immunity and metabolism. And I literally will spend one slide summarizing the work in the hemovascular or immune system, 20 years worth of work in one slide, metabolism on a second slide, again, about 20 years worth of work, and then just share with you a recent story on KLFs, myeloid cells, and immunothrombosis. So with that, I'm a cardiologist by training, and so I have thought a lot about the circulatory system. And when I was in Dr. Haber's lab, so this is mid to late 90s, we were working on issues that were very important in the day, things like transplant arteriopathy, atherosclerosis, restenosis. And so that gave me my initial entree into molecular biology, or a relatively new field at the time, molecular cardiology. And as I matured in my 3-year stint with Dr. Haber in his lab, started to think about the vascular system a little bit differently, if you will, almost from an evolutionary perspective. And if you think about it, the vascular system as we know it today, a closed vascular system, actually came into existence about 600 million years ago. And it's a really interesting time period because blood cells and a closed propulsive vascular system arose around the same time and was absolutely fundamental to the ascension of multicellular or Metazoan life, because you could now deliver oxygen and nutrients at a distance. And coincident with that time period was increase in the rise of atmospheric oxygen levels. So hold on to that set of thoughts. Second, if you think about what blood vessels do fundamentally, it is to deliver oxygen and nutrients, i.e. metabolism, and deliver defense at a distance, immune cells, immune factors, et cetera, that are in your bloodstream. Deliver them for the survival of the organism. Survival because you can produce energy, metabolism, and defense. And again, as I was finishing up in the Haber lab, and I was going to be doing my clinical cardiology, but what I was really intrigued by at the time, this is late 90s now, was if nature were parsimonious, would there be common conserved pathways that would coordinate the blood, vascular, and metabolic systems, all that had common origins, certainly temporally, 600 million years ago, and also because if, for those that know this space, precursor cells called the hemangioblast gave rise to blood and vascular systems. So it was these kind of concepts, and could there be conserved pathways that we could identify? Through a series of, and I won't bother you with sort of the details, but we undertook a very old strategy called differential display from that nobody does today, and cDNA homology screening, which is just a very painstaking approach to discover new factors. But we stumbled upon a family of factors that were expressed in vascular cells, immune cells, and in metabolic cells. And it happened to be in a family of proteins called crupples. So what are crupples? So cruppel in Drosophila is actually a really famous gene. It was identified in the 70s and 80s by Nusson Volhard and Eric Weishaus, and there's a paper from them shown for you here. And it's a DNA binding protein that regulates Drosophila development. And that concept of a genetic factor, molecular factors governing development was very central to those two individuals, along with Ed Lewis, receiving the Nobel Prize in Physiology and Medicine in the mid-90s. So that was interesting. They seemed to be important, but that was Drosophila. And we identified these factors in mammalian tissues. So was there much known about mammals? Well, at the time, not really, but there was another interesting piece of data. So this is a paper, it's a single author paper, and it showed that the Drosophila cruppel, when the fly became an adult, was only expressed in one region of the fly, in what's called the fat body. And the fat body of the fly is really interesting because it basically contains three tissues: blood vessels, immune cells, and metabolic cells, the adipocyte or the hepatocyte equivalent of the fly. So remember, we were looking for factors that would be involved in blood vessels, immune cells or blood cells, and metabolism. How interesting that in this little organism, the fly, in the adult fly, it was expressed only in the place where there were blood vessels and immune cells and metabolic cells. And now I'll just speed up a little bit, but I wanted to set the stage and share with you that it's conserved across phylogeny, across Metazoan phylogeny, from flies to mammals. We discovered a number of these KLF factors, and now there's many of them that have been identified, a total of 18 in mice and in humans. And there are a number of family members that we and others have discovered that regulate blood vessels, blood cells, immune cells in particular, and metabolism. All right. So in the next two slides, I'm going to summarize 2 decades of work and then go into the principal story that I wanted to share with you today. The field of KLF's controlling metabolism began with our paper. It was actually my first senior author paper in JBC in 2002. And I'm not going to bore you with the details, but at a high level, what I can say today, now fast forward over 20 years, is that members of the KLF family regulate how we acquire nutrients, how we utilize nutrients, how we make energy out of nutrients across all major nutrient classes, glucose, lipids, amino acids, and proteins, that they regulate the power plant in the cell that makes energy, the mitochondria, and the mutations have been identified in these factors that regulate metabolism. And you can see this is a subset of the papers that we have published. And in particular, this cartoon in the middle shows, it conveys to you that we have used the physiology of the fasting response, endurance exercise, and day-night cycles to glean these insights. Again, that's not the story we're going to talk about today, but I'm happy to talk to anyone afterwards if you're interested, or you have some of these references should you wish to get more information. I'm going to summarize now again about 2 decades of work on KLFs as essential regulators of the blood vascular or hemovascular system. And what we discovered is that KLFs are nodal regulators of the production, essentially, of all blood elements. Endothelial cells actually, and all blood elements have a common progenitor, and they regulate all of that. So they regulate the endothelial cells of blood vessels and all major blood cells, red blood cells, platelets, and the innate and adaptive immune systems. And mutations that have been identified now in a number of disease processes. So this was just sort of the background, and I just want to make sure Sharon Oaxaca and I had talked about a natural inflection point. That's just sort of setting the foundation for the main story, and I wanted to make sure if there were any questions, I'm happy to take them on the background of KLS before I launch into this recent work.
Let's see, I'm looking in the chat. I don't see anything.
Okay.
So proceed.
Proceed. Okay. All right. Thank you. All right. The study that I'm going to share with you is a recent publication from 2 fabulous individuals. The person on the top, Lalita Nayak, she is a hematologist oncologist that trained with me, now has her own independent lab at Case Western. And the young man below her is David Sweet, one of the brightest young physician scientists I've ever met. David did his MD PhD with me. He's graduating from Case Western and is currently applying to internal medicine programs. And the focus is going to be on KLFs in the hemovascular system, with a particular focus on myeloid cells, so macrophages and neutrophils. All right, so we're going to be talking about thrombosis. That involves the circulatory system, obviously. And this graphic is just a reminder for me to tell you that heart and circulatory disorders are still the leading cause of death and disability. And a strong case can be made, if you think about it, that diseases of the circulatory system that affect everything from the top of your head to the tips of your toes, shown for you on this diagram, are actually the leading cause of death and disability worldwide. So having a greater understanding of vascular disease is of obvious importance. All right, we're going to be talking about blood clotting and thrombosis. And so I just want to deal with this issue of hemostasis versus thrombosis up front, and then we'll get into the data. Hemostasis is a physiologic process, absolutely requisite for survival. So when you cut yourself, you obviously want the bleeding to stop. And that is the process of hemostasis. And it involves your endothelial cells of your blood vessel, platelets, and coagulation factors. And these elements work together to form a little plug that prevents you from bleeding out. Very important. And there are a number of pathways, platelets and clotting factors working together with your endothelial cells. And these are some of the major pathways involved in making this little plug that prevents you from bleeding out. Thrombosis is not a physiologic process. It's gone awry. And I don't, you can consider it a hemostasis gone awry, but there's a really important difference between hemostasis and thrombosis, which is it involves the same cellular players with one addition, immune cells. And that's what this cartoon is seeking to convey for you. Immune cells, particularly myeloid cells like macrophages and neutrophils. And that's really, whoops, sorry about that. That's really important because if you think about all the therapies that are used clinically today, they actually impact hemostasis, the physiologic process. And if you think about it, that physiologic process, if you intervene, yes, you will reduce blood, you will impact thrombosis, but it makes sense that a side effect of that would be bleeding. And that's why clinically, many of you know, we're always, when we give these agents, we want to reduce blood clots, but we always worry about bleeding as an ill consequence of these therapies, and sort of balancing that risk of helping alleviate the thrombotic burden, but at the same time, balancing that with bleeding risk. And if you accept this simplistic view that I just shared with you about hemostasis and thrombosis, you might say, well, since there's a distinction between these two processes centered around these immune cells, wouldn't it be great if we could target immune cells? And so I'll ask you to just keep that in mind as we go through the rest of the presentation. The disease process that we're going to focus on is a really devastating thrombotic condition called antiphospholipid antibody syndrome. I should have spelled this out for you. I apologize for that. Antiphospholipid antibody syndrome, APS for short. And it is one of these really devastating conditions, often comes to light clinically in young women, particularly women of childbearing age who have frequent failed pregnancies. And that is because there is an abnormal clotting process that can occur that renders the fetus non-viable. Antiphospholipid antibody syndrome is an autoimmune disorder that's associated with both arterial and venous thrombosis. So that's very interesting, important, and obviously clinically problematic. Despite our blood thinners, whatever we might use, and I showed you some of those on the previous slide, recurrent thrombotic events still continue. Studies to date have focused on platelets, endothelial cells, and because of the loss of the ill consequences during pregnancy, a number of studies have focused on trophoblasts. But if you think about what we just discussed on the prior slide, what about the role of the immune cell? The work that I'm going to share with you, a lot of it is done in mice, and there's going to be two assays that I'll reference, and so I just want to explain them to you up front. In mice, we can look at arterial thrombosis and venous thrombosis, and there are a number of different ways to do this. The one that we used in the arterial side is called the carotid artery laser injury model, shown for you here in a bit more detail. But basically, you give a dye, a photochemical dye called rose bengal. It's injected into the mice. You shine a laser. It leads to, it's sort of a redox injury of the endothelium, and blood starts to clot. And you measure the time to thrombotic occlusion. So you cause the injury, and typically in a mouse, it takes a good 20, 30, 40 minutes, depending on the situation, for complete arrest of blood flow in the carotid artery. And that's what you're measuring, time to occlusive thrombus, as we like to call it. The shorter the time, the more thrombotic the animal. So if it takes you 40 minutes to occlude blood flow, and then in condition number 2, it's 20 minutes, that's would be interpreted, the 20 minutes is more thrombotic because it's occurring faster. The opposite, the other assay is a venous thrombosis model where we put a ligature around the IVC of the mouse, and of course there's stagnation of blood flow, Virchow's triads for the aficionados in the audience. And the readout is how big, how heavy is that clot? The heavier the clot, the bigger the clot, the more thrombotic. So on the arterial side, the shorter the time to thrombosis, the more thrombotic you are. And on the venous side, the heavier the clot, the more thrombotic you are. All right. So the first thing we did was we took antiphospholipid antibodies from patients who have antiphospholipid antibody syndrome, and we determined if we give those to a mouse, does the mouse become more thrombotic? So here's a mouse, a wild type mouse, WT is wild type, and we give these antibodies, remember these are from humans, and then we do our injury, and I'm going to just focus on the arterial side, but the venous data is concordant. And you give this antibody, and then you look at the time to thrombosis on the arterial side. So here's a normal mouse with a control IgG, and it's about 40 minutes. Very normal for a C57 mouse. When you give the antiphospholipid antibody, APL for short, the time is shorter, about 20, 25 minutes. So this mouse, once the one that received the antiphospholipid antibody, is more thrombotic. So just like the human condition, the antiphospholipid antibodies, when given to the mouse, confer a more thrombotic state. We then did a couple of different manipulations. So there are ways where we wanted to focus, remember, on the myeloid compartment, that's macrophages and neutrophils. And we were curious, are macrophages important? Are neutrophils important? Or are they both important? Not shown for you on this slide is data showing that if we deplete macrophages, there was no effect. The mouse was still thrombotic. But if you deplete neutrophils, and you can do this with an antibody called anti-Ly6G, details not important, it's a very standard approach. So if you deplete neutrophils in a mouse and you give them this antiphospholipid antibody, you reverse, you overcome the thrombosis. Here's a mouse that received the antibodies. Here's a mouse that received or was depleted of neutrophils. And you see the time, which was around 25 minutes, is now back up to nearly 40 minutes. And we did other experiments. I won't go into the details, but where you can take neutrophils that have been exposed to antiphospholipid antibody, transplant them into another mouse and see if it confers a prothrombotic effect. And can you reverse that with neutrophil depletion? All of that and a series of other, corroborative studies were performed. But the take-home message is, one, antiphospholipids from patients when given to a mouse confer arterial, and I didn't show it to you, it's in the paper if you're interested, arterial and venous thrombosis, and neutrophils are critical. And there was minimal contribution of macrophages. So we walk away from this set of studies that neutrophils are critical for the thrombosis seen in antiphospholipid antibody syndrome. Why would this occur? What is it about the neutrophils? So we took neutrophils from patients with antiphospholipid antibody syndrome, and we did what's called RNA-seq. This is a way for us to look at the entire RNA profile of normal neutrophils and neutrophils from patients with antiphospholipid antibody syndrome. And one of the genes that was most markedly reduced in the neutrophils of patients with antiphospholipid antibody syndrome was one of our favorite factors. So how lucky is that? It's called KLF2 or Krupa-like factor 2. And if you give antiphospholipid antibodies to mice, their levels of KLF2 also go down, not as dramatically, to be honest, as the human, but still in a significant manner. So this was exciting to us, and we thought, wow, at least there's a connection between a factor that we know well, we helped discover some years ago, and this devastating condition, antiphospholipid antibodies syndrome. So we then created a mouse that was deficient in KLF2 in myeloid cells. So that's really exciting. We have a mouse now. You have control mice. They're called lysozyme M-CRE, but I'm just going to call them control. And you have mice that are myeloid deficient in K2, K4. So they're deficient, truth be told, in both the macrophage and neutrophil compartments. So that's a little bit of a caveat to these studies, and I'll show you how we got around that. But just important for you to know that. So we made these mice and we subjected them to our two favorite assays, arterial and venous injury. These are control mice, about 40 minutes in arterial thrombosis, time to occlusive thrombus. And the myeloid KLF2 knockout mice are at about 24, 25 minutes. So they are prothrombotic. On the venous side, remember, this is where the bigger the clot, the more prothrombotic you are. And sure enough, these myeloid K2 mice have bigger clots. So arterial, shorter time to thrombosis, venous side, bigger clots, they're prothrombotic. Great. And we went on to show, and I'm going to fast forward a little bit and ask you to trust me with the interpretation of the data and some of the published work, but that's not shown for you on this slide. But we went on to reassess now in the myeloid KLF2 knockout animals. Remember, KLF2 is gone in both the macrophages and neutrophils. We wanted to confirm, is it the neutrophil KLF2 that's important? And we did the depletion experiments. We did, can we confer a prothrombotic phenotype just using neutrophils alone that are deficient in KLF2? And we looked at both arterial and venous thrombosis. And the conclusion was that KLF2 deficiency just in neutrophils confers a prothrombotic phenotype. Great. Why? Why should loss of KLF2 in neutrophils lead to more thrombosis, both on the arterial and the venous system? Well, there's a lot of information about how neutrophils might be involved in thrombosis. And that has to do with their ability to extrude things called neutrophil extracellular traps. I'll describe that on the next slide in a little bit more detail. They can produce and release things like tissue factor, which is a really powerful prothrombotic factor. And perhaps the loss of KLF2 in neutrophils allows these neutrophils to be more aggressive in adhering and migrating and coalescing around a site of injury to cause thrombosis. And we explored all of that, and I'll go through this relatively quickly and give you the take-home message. Neutrophil extracellular traps, for those that may not be familiar, these are net-like structures shown for you in this graphic, composed of DNA, histones. This is the stuff contained in the nucleus of the neutrophil, and it can be spit out, extruded out of the neutrophil. It's thought to actually be a defense mechanism, but when it goes awry, it can lead to thrombosis. And there's ways that you can stain for these neutrophil extracellular traps in neutrophils and see, are they producing more or less? And that's, again, for the aficionados, you can stain with citrullinated histone. Details not important, but I think you can see that compared to control neutrophils in a Petri dish, knockout neutrophils, that's what K2KO means, knockout of KLF2 in neutrophils, there's more green staining, suggesting that they produce more neutrophils. And we confirmed that was in vitro, and we confirmed that there's more histone, citrulated histone staining in vivo in clots as well, be it arterial or venous in mice. We did a lot of mechanistic studies for those that are interested. We know that neutrophil traps are formed through the actions of a number of enzymes. They're called myeloperoxidase, neutrophil elastase, etc. And we showed that in the absence of KLF2, the levels of some of these factors, these enzymes that produce neutrophil extracellular traps, are elevated. You can see neutrophil elastase here, NPO levels are higher, and this is just quantitation of that. Remember I said there could be, perhaps there's more tissue factor, and indeed in knockout neutrophils, there's more tissue factor at the protein level and at the activity level. And then finally, we all know that one of the big functions of neutrophils is wherever there's injury or infection, they hone into that area, they adhere to endothelium, they migrate across the vasculature to, respond to an injury. And we wondered, are these neutrophils that are lacking KLF2 somehow more aggressive in their adhesive and migratory capabilities? We had some hints of this because when we did our RNA-seq, again, looking at the transcriptome from normal neutrophils and neutrophils deficient in KLF2, we saw a number of pathways related to immune activation, no surprise, but also to adhesion, migration, and actin remodeling. I'll get to that in a second. But we knew there was something going on with adhesion and migration, and both in vitro studies, in vivo studies, and in vivo studies looking at blood vessel injury, arterial thrombosis or venous thrombosis, we could show in a time-dependent fashion that the neutrophils get to the site of injury and they start to form the clot faster and more aggressively. So to summarize, remember we were looking at nets, we were looking at tissue factor and adhesive and migratory capacity. All of those were heightened in the absence of KLF2 and neutrophils. So we have at least some mechanistic understanding of why loss of KLF2 would lead to bigger clots, arterial and venous. Now, we wanted to explore, and particularly David and Lalita wanted to explore this a little bit more. And they started to think about neutrophil migration and adhesion, which many of you will know is a really precisely coordinated process. There's a leading edge and a back edge. This is, again, for the experts in the audience. But what's really interesting about neutrophil migration and adhesion is that there's all kinds of remodeling of the cytoskeleton that occurs below the cell membrane that promotes clustering of adhesion molecules in areas called uropods, that's shown for you here. And it actually anchors the cell as its leading edge is moving. And because there was actin remodeling, remember that RNA-seq pathway I mentioned? There was actin remodeling along with more adhesion migration. Lalita and David began to wonder, is there evidence of actin remodeling and this uropod-like formation that is heightened in neutrophils that are deficient in KLF2? And so they did the staining for neutrophils that are activated in a petri dish, and indeed there is more actin staining. That's what that green line is compared to this one. You can even see these protrusions that are really indicative of actin remodeling. And you see the same thing, slightly different color scheme here using confocal in neutrophils from patients, directly from patients with antiphospholipid antibody syndrome. Look at just the thickness of this actin staining and this one in the APS patient. So loss of KLF2 or exposure in patients to antiphospholipid antibodies leads to cortical actin reorganization. There's reorganization, and then, but what makes them more sticky? What makes them adhere more vigorously? And we used classic flow studies where you can flow neutrophils across a chamber, and you can coat the chamber with classic adhesion molecules that are expressed on the endothelium and known to grab neutrophils out of the circulation. And some of the major players that do this are called P-selectin or VCAM, ICAM. And so in a series of studies where I'm going to give you the punchline, we undertook, we're watching neutrophils flow over these chambers coated with these different adhesion molecules. And the bottom line, or the take-home message from these studies, are that knockout neutrophils adhere more vigorously to P-selectin. They don't really care about VCAM or ICAM, but somehow these neutrophils deficient in KLF2 are really, really primed to stick to an endothelial factor called P-selectin. You might say to yourself, that's nice, why should I care? The reason you should care is because on neutrophils, there's a factor that usually aggregates in those uropods called P-selectin, that binds P-selectin, called PSGL1. So we thought to ourselves, oh, this is going to be really easy. The neutrophils that are lacking KLF2 stick to P-selectin, so they must have more PSGL1. Mystery solved. But sadly, there was no difference in the levels of PSGL1. But what we did see is in an activated neutrophil that the PSGL1 would aggregate in those little uropod structures that exist on neutrophils. And that's really interesting because we thought to ourselves, if the PSGL1 is aggregated around activated neutrophils, could we target PSGL1? If we could do that effectively, and if it's clustered on these uropods, an agent can get at activated neutrophils and leave activated neutrophils alone, could that be a therapeutic opportunity? So the hypothesis is shown for you here. Normally, neutrophils do have some PSGL1 distributed all over the place because they're always looking to see if there's anything bad going on because they need to stick to the endothelium. But when they're activated, the PSGL1 is sort of, through this actin remodeling, localized to the uropod to allow for aggressive attachment. And we thought to ourselves, if we could intervene, particularly in these activated neutrophils, somehow by blocking the ability of the neutrophil to bind to endothelium, you might be able to avert the thrombotic effect. Kind of a simple idea. And we worked with some of our biomedical engineer colleagues who developed a nanoparticle-based system to block neutrophils. And so I'm going to walk you through this here. These are control, and this is looking at cells attached in those flow assays. We're flowing knockout neutrophils and 1,000 cells attached. If you use a simple antibody that blocks neutrophils, it has some, blocks PSGL1, it has some effect. But if you use a nanoparticle that's coated with this antibody, it actually has a more aggressive effect because you can intensify the blockade of PSGL1 on the uropod, and you get a heightened reduction in adhesion. Okay. And then we thought, okay, if this works, we develop these nanoparticles, can we inject them into mice that are deficient in myeloid KLF2 and rescue arterial and venous thrombosis? And indeed, that's what this bottom part shows, that if clotting, which occurs in 20 minutes on the arterial side, with these nanoparticles, you can basically restore it to normal times, 40 minutes. And on the venous side, remember here, smaller is better. If you give these nanoparticles, the clot that weighs 40 milligrams is now less, 28 milligrams or so. And then finally, this is done in KLF2 knockout mice. What about that initial experiment? Take a normal mouse that you give the human antibody to. Now, after you give the antibody that causes thrombosis, the antiphospholipid antibody, you actually treat them with this nanoparticle. Would you prevent arterial and venous thrombosis? And obviously the answer is yes, otherwise I wouldn't be showing it. Again, 20 minutes because of the antibody. This is not knockout. These are normal mice now receiving the antibody, and you restore time to thrombosis to normal on the arterial side, and you make the venous clot smaller. And there was no effect on bleeding time. Remember, you want to balance thrombosis with bleeding, and there's a classic assay that you do called tail vein bleeding time, which was completely unaltered between animals given no nanoparticles or those given nanoparticles. And so this is the conclusion, summary diagram. In antiphospholipid antibody syndrome, there's a reduction in KLF2. It leads to net formation, tissue factor release, actin remodeling, and we've taken advantage of the uropod PSGL1 p-selectin interaction to try to interrupt immune cells sticking to the endothelium for therapeutic gain. And this is part, the myeloid control that KLF2, or the control that KLF2 has on the myeloid compartment broadly is much more expansive than just thrombosis. That's what I shared with you here. But we have published or unpublished work that it also impacts metabolic disease, brain health, which is linked to vascular function, and a number of other age-associated disorders. And that's work, some published, a lot of it unpublished, that we hope to pursue here. And so with that, this is my final slide before a few thanks. Overall summary of the KLF field. I began with immunity and metabolism, or the hemovascular system and metabolism, nodal regulators. They're altered in disease. They are potentially, or the studies of KLFs have the potential to lead to therapeutic opportunities. And the work, I think now 20 years later, suggests that they're important regulators of age-associated disorders, and in work that I did not present to you at all, but actually important in the very process of aging itself. Again, that's a story for another day. And the most important slide, just thanks to a lot of trainees over the years, I think now had 39 trainees, Sharon, that now grace the faculty of institutions across the land and globe, and our funding sources, and of course, by my very humble lab at Brown today. So with that, I'll stop, and we have a good 10 minutes or so, Dr. Rounds, for questions.
Fantastic. Thank you so much. This is really terrific. So we have a question from Ronnie Elwee, who notes that she's out of her lane here as an implementation scientist, but she has
implement nanoparticles in people. Yes, no, it's right up Ronnie's alley.
She has some great questions. How does a clinician recognize KLF2 deficiency?
Oh, it's a great question, Randy. So we have been working, and I referenced this young investigator, Lalitha Nayak. I should say, more importantly, I shouldn't say we, I would say Lalitha has been working very hard and has made really good progress on developing a rapid PCR-based diagnostic test for KLF. So that would be really exciting. And number two, extending that, Ronnie, there are mutations that have been identified that lead to mutations in KLF that have been associated with cancer and other prothrombotic disorders. Cancer is one. And so she's not only developing the diagnostic to measure KLF2 levels in circulating cells, but also, a laboratory test that you could send off to look for mutations akin to, looking for protein C, protein S deficiency, or factor V Leiden mutations. So great question and efforts that are in the works.
And then Ronnie goes on to ask as to whether this work has progressed to developing guidelines for clinicians.
Oh, no, I'm a few.
You're decades off.
We are some time away, Ronnie. The nanoparticle-based therapeutic, nanoparticle-based therapy have entered patients. So that is a possibility. But there are a lot of issues related to pharmacokinetics and toxicity and other that still need to be worked out. So the short answer to your question is that we are, frankly, a few years away from entertaining getting into people, let alone changing guidelines.
Okay. So Gaurav Chowdhury asks, would targeting P-selectin interaction with neutrophils perhaps increase the risk and severity of infection?
Superb question. The answer in principle is yes, because neutrophils are so important in our defense mechanism and they leverage PSGL-1 for that. The one thing I would say is we're thinking, Garv, that in diseases like antiphospholipid antibody syndrome, the neutrophils are activated and you have a thrombotic event. If you could intervene for a finite period of time, right? And I don't know, Sharon, you probably took care of APS patients through the course, right? They come in and it is dramatic, right? Arterial and venous thrombosis, pregnancy loss, maybe, and clots all over the place. I think there's an opportunity, Gaurav, to use the lessons learned from these observations in the acute setting to overcome the thrombotic risk. Long term, if you could target the uropod, and I don't know if you would do this long term with nanoparticles, my instincts are that a small molecule would be better, but if you could somehow target an activated neutrophil and not a normal neutrophil running around, you might be able to get the beneficial effects without the expected impact on infectious, or increase in infection. That's at least my thinking. Again, that's why the targeting of PSGL-1 just on that little uropod in an activated neutrophil is so important versus all neutrophils running around.
So related to that, I would ask, would targeting the uropod, how would that affect phagocytic function of the neutrophil? Because there's certainly actin remodeling with phagocytosis, such as bacteria, for example.
Yeah. So I can tell, I don't know if I'm going to be able to give a great answer, but let me just share. One, I'm not sure exactly how targeting the uropod affects phagocytosis. I don't know that. In previously published work, we have shown that loss of KLF2 augments phagocytic capacity. So that is true. That was a paper in immunity now a few years ago, Sharon. So there is enhanced phagocytic capacity. That shouldn't surprise you, I think, based on what I've shown, because the neutrophil is clearly more activated and adheres and migrates more vigorously. But it's a very good question that I don't know the answer to, which is when you target the uropod, let's say like with these nanoparticles, we did not do studies in that context to see how it affects phagocytic capacity, being able to kill bacteria in a petri dish or in a, there's models of infection like the oh God, I'm blanking on it, but where you puncture the bowel of a mouse and have a colitis type model. We have not done those studies. That's a good question. Don't know.
And then another question is, there are conditions where in situ thrombosis is observed. So one is, pulmonary arterial hypertension, acute respiratory distress syndrome, lung disease I'm familiar with. And have KLFs been studied at all under those circumstances? I'm not aware of any. It might be an interesting area.
A former trainee of mine, Ann Hammock, published some work demonstrating gain and loss of function in an endothelial, so where you manipulate the KLF in the endothelium.
Okay.
And using a hypoxia model of pulmonary hypertension, that it alleviates pulmonary hypertension. In situ thrombosis, I'm not aware of an experimental model where that is seen, like in the, I don't know in whether it's mice or rats. I don't know if you typically see in situ thrombosis, but regardless, I know Ann did not look at that. But the animals in the absence of KLF are definitely more prone to pulmonary hypertension.
Oh, wow, interesting.
It was published in the, what is that, you guys, the pulmonology, the Red Journal, is that right?
American Journal of Respiratory Cell Molecular Biology.
Yeah, that's it. Yeah. And I can get you the reference if you're interested, but that's the one paper that I can think of, happens to be by someone that trained with me, so I know.
Okay, great. Well, I don't see any other questions in the chat. I want to thank you once again, Mukesh. This was it's beautiful work and so very clearly presented that even our implementation scientists are starting to think about KLF. So,
thank you, Ronnie. Thanks, Aaron.
Bye-bye.
Bye.